Here's some definitions that we need to understand to have mastery over how inheritance works. The first term is phenotype, which is all of the observable traits of an organism. The phenotype may or may not match the genotype, which is the genetic makeup of an organism. Homozygous refers to the fact that an organism carries two identical alleles for a particular gene, and heterozygous means that we have two different alleles for a particular gene. If we look at these flowers on your screen, we can see some purple flowers and a white flower, and notice that the phenotype ratio does not match the genotype ratio based upon the rules of inheritance. If the organism is homozygous, meaning they carry two identical alleles for a particular gene, then we can see that the flower is either purple if they're homozygous dominant, or white if they're homozygous recessive. However, if they are heterozygous, they are going to be a purple flower because they carry a copy of the dominant and recessive allele, but the dominant allele is the one that is expressed. Homologous chromosomes are the pairs of chromosomes in a diploid organism, and they pair up during meiosis. And also remember the definition of alleles, these alternative or variant forms of our gene. Alleles are said to be recessive if the organism must have two copies of the allele to see a result, and alleles are said to be dominant if the organism only needs one copy of the allele to see the result. And notice that the genes and those versions of those alleles are found at loci, which are located along our homologous chromosomes. The traits that these alleles are responsible for have different degrees of dominance. The first degree of dominance is complete dominance, in which the phenotypes of the heterozygote and the dominant homozygote are identical. The second degree of dominance is incomplete dominance. In this example, the phenotype of the heterozygote is intermediate between the homozygotes. And the last example is codominance, which means two different alleles affect the phenotype in separate but distinguishable ways. Remember, phenotype is the observable trait. Let's look at an example of a single autosomal gene which provides the blueprint for the widow's peak hairline. We could label our alleles as either big W, which is this uppercase W for widow's peak, or little w, which is a lowercase w for a straight hairline. We're saying that widow's peak is inherited as a dominant condition. So in your genes, if you have both copies of the dominant allele, which is the uppercase W, you have a widow's peak. If you are heterozygous, meaning you have the big W and the little w, you have a widow's peak. It's only if you have both copies of the recessive allele, which is little w, that you have a straight hairline. It's important to note that autosomal traits, such as hairline, are not sex-linked. Instead, these are carried in our autosomal cells. Here's an example. You can practice doing these crosses. If mom has little w, and dad has little w, meaning they both have a straight hairline, then 100% of their children will have a straight hairline. But if mom is heterozygous and dad is dominant homozygous, meaning he is carrying both copies of the dominant W allele for widow's peak, the children are going to be 50-50. Half of the children will be like mom, and half of the children will be like dad. The way that we complete these crosses is with a special diagram called a Punnett square. Codominance is another way that we can have a degree of inheritance, and an example is in sickle cell anemia. Here we can see normal blood cells next to sickled blood cells, and we have different versions of a single gene that are going to be codominant for these two shapes of cells. In your genes, if you have the uppercase S, both copies of the dominant allele, then you have normal red blood cells. If you carry both copies of the recessive allele, the little s, you have sickle cell anemia. But check out the heterozygous individual. The heterozygous individual has the big S and the little s in their genotype, which means they have some normal red blood cells and some sickled red blood cells in their blood. That's why we call this codominance, because both are expressed side by side. This actually gives some populations a selective advantage. For example, we find this sickle-celled codominant trait in 1/10 of African Americans. If sickle cell anemia is such a bad disease, then why would one population in our country tend to have so many of these genotypes? Well, the short answer is it provides ancestors a resistance to malaria. So if we look at this global map, we can see all the places where malaria is found on our planet. And in particular, there's a lot of regions in Africa that have malaria. And if you have some sickled blood cells, because you are heterozygous for sickle cell anemia, you will be a healthy individual, but you will have resistance to this blood-borne pathogen. Codominance also plays a role in our blood types. In codominance in blood cells, more than one allele encodes for a functional protein found on the surface of that blood cell. If two dominant alleles are present, both proteins encoded by those alleles will be represented in the phenotype. Is this sex-linked? No. Remember, there's no X or Y chromosome here. We're just talking about these autosomal characteristics. In human blood types, we've got both A and B as dominant alleles. Genotype AB confers red blood cells with both A and B molecules on the surface of that red blood cell. However, we also have another gene, so this is multiple alleles that are being controlled and expressed in different ways. The I gene also has a recessive allele, which is represented by lowercase I, and this encodes for a non-functional protein on the surface of our red blood cell. But the two dominant alleles, both A and B, make the I gene codominant. People with type O blood have no proteins on the surface because they carry both copies of the recessive allele, which is noted by little I and little I. People with type O blood are considered to be universal donors because anyone can take their blood in a blood transfusion. Now, we can determine how alleles and genes will be inherited if we do a Punnett square. Let's say that our kitties have these two genotypes. The white kitty has a big B, and the black kitty has little b, little b. When we do a cross, every one of their babies has a big B, little b genotype, which is to say they are all heterozygous for this fur color. Therefore, 100% of the babies will be white kitties. But let's say fur color is inherited codominantly. That means a little bit of white fur and a little bit of black fur are expressed. Therefore, 100% of our babies will have a little bit of white fur and a little bit of black fur. It's important to note that some traits are controlled by multiple genes. In fact, many traits are controlled by multiple genes. To give us an example, here's the pugil. The pugil is half pug and half beagle. But if you mate a pug with a beagle, you're not guaranteed to get a pugil because many different genes control the physical appearance or the pugilness of our pugil puppies. We can't just track versions of one single gene. We need to actually track the chromosomes because remember, independent assortment and crossing over are always occurring. This is all an example of polygenic inheritance. Our environment also shapes the physical appearance of organisms. Remember that genes provide the blueprints for proteins, but things like nutrition, exercise, sunlight, and social environment all lead to our development and can change our appearance as well. And developmental conditions are not things that we can inherit. What other factors affect skin tone then? Well, if the phenotype is a combination of genes plus environment, we can also include diseases, which are going to be a result of genes plus environmental factors. Here's our next question. In humans, the sex cells, which are the gametes, are produced by which of the following? Independent assortment, mitosis, meiosis, crossing over, or are all of the above correct? Pause the lecture if you don't want to hear the answer yet. Meiosis is the answer. Sex cells are a result of meiosis, and we will see why in just a minute. In humans and some other animals, there is a chromosomal basis of sex determination. The sex chromosomes have genes for many characters unrelated to sex. A gene located on either sex chromosome is what we call a sex-linked gene. In humans, sex-linked usually refers to a gene on the larger X chromosome. Here we can see a male and female with 44 plus XY chromosomes, and the female has 44 plus XX chromosomes, right? So that's a total of 46. And then when we make our gametes, we cut those numbers in half, and therefore sperm can either carry X or Y, and the egg always carries an X. Therefore, we have a 50-50 chance of getting a male or a female baby. Chimpanzees are our closest living relatives. They have been on a separate evolutionary path from humans for only 6 to 7 million years. Subsequently, we still share most of our genes with chimpanzees. We differ by only 1 to 2% in terms of DNA sequence. However, the genes on the Y chromosome are a major exception. The DNA sequences on Y chromosomes of chimpanzees and humans differ by almost 30%. This indicates that Y chromosomes have been evolving at a much faster rate than the X and all other chromosomes. Sex-linked genes were discovered during studies on fruit flies many years ago because they were trying to figure out how eye color was inherited in these flies. Over 1,000 genes are linked to the larger X chromosome, but only about 26 genes are linked to the Y chromosome, including the gene that is responsible for the development of testicles. Some of the diseases that can be inherited by sex-linked disorders includes red-green color blindness, hemophilia, and Duchenne's muscular dystrophy. Sex-linked genes have unique inheritance patterns based upon if they are linked with the X or the Y, but remember most of them are linked with the X. And those X-linked recessive disorders affect way more males than females. Females must receive a recessive allele on both X chromosomes to express an X-linked recessive disorder. Males only have one X chromosome. To express a recessive disorder, they only need to inherit one X-linked recessive allele. Here's a red-green color blindness chart, which doesn't quite look as good on your screen as it does when you look at it in a doctor's office, but this is how they can help determine if you might have red-green color blindness. Color blindness is characterized by a malfunction of light-sensitive cells in the eyes. If you have no color blindness, then on your screen you can read 25, 6, 8 across the top, and 56, 29, 45 across the bottom. Hemophilia is another genetic disorder that is sex-linked, and we see a lot of this in the royal families because the royal families of Europe have been historically quite inbred. Hemophilia is a disease that means your blood doesn't clot normally, and when we see how disease is passed on through the generations, we look at a family tree that is specifically called a pedigree. All right, here's our another challenge question. Why are sex-linked disorders more common among men than women? Your first choice is mutations usually occur on genes associated with the X chromosome. Next is mutations usually occur on genes associated with the Y chromosome. And your last choice is men are more likely to get the dominant gene that carries a disorder from their mother. So pause the lecture if you don't want to hear the answer yet. The answer is A. Mutations usually occur on genes associated with the X chromosome. It's important to note men are not more likely to get the dominant gene with the bad allele from their mother. A female is just as likely, but the female will likely have the other X chromosome to protect her from the effects of that bad allele. Now let's look a little bit at the history of how inheritance was first hypothesized. We credit Gregor Mendel as the father of genetics, even though back then he had no idea what DNA even was. DNA had not yet been discovered in the 1800s, but Gregor Mendel did understand how inheritance worked. He actually used pea plants as his model organism, since those plants are easily manipulated. There are many advantages to using pea plants for genetic study. There are many varieties with distinct heritable features or characters such as flower color. There's many variants for these traits of these genetic characters. The mating of these plants can be easily controlled. Each pea plant has both sperm-producing organs, which are called the stamens, and egg-producing organs, which are called the carpels. Cross-pollination, which is that fertilization between different plants, can be achieved by a human by simply dusting one plant with the pollen from another. That is why Mendel used these pea plants so that he could do his breeding experiments. These are just some of the traits that he was able to identify. There are 7 traits listed here, each expressed by alternate versions of the same gene. In summary, Mendel had 4 hypotheses, and we're going to use the modern terms of genetics and apply them to these hypotheses. Number 1, there are alternative forms of genes called alleles. Number 2, for each characteristic, an organism inherits 2 alleles, which is to say 1 from each parent. Number 3, alleles may be dominant or recessive. And number 4, gametes carry only 1 allele for each inherited characteristic.